I'll speak about quality aspects in the IMP developments. The structure of this speak is presented here. I would like to speak about the importance of relevant quality information in a clinical trial application. And I would like to stress the different phase-dependent requirements for quality documentation in clinical trials. There is some EU guidance on the quality requirements. There's a guideline, and there is some questions and answers, and I will cover this. And I will also speak about the relevance of keeping a future MA application in mind already at the clinical trial stage. And in the last slides, I would like to give some examples. The quality documentation can also be, some people call it the CMC. I think this abbreviation is for chemistry, manufacture, and control. And just to recapitulate briefly, it's about the nature of the drug substance and the drug product, and the manner by which they are made, and the manner by which the manufacturing process is shown to be in control. I'd like to stress that the quality documentation is equally important as other parts of the submission. And in the inclusion of this type of this chapter of documentation became a European requirement in 2004 by some guidance given by the European Commission. Before that, some countries had their own practice, but it was not a common European requirement before that. The quality documentation is the basis for drawing conclusions from the clinical trial, and it links to the future MA application. And therefore, the quality documentation needs to be of a certain standard in order to fulfill this. And in order to avoid major objections to the clinical trial documentation during the assessment, the applicants should try to include the relevant documentation when the application is submitted. And otherwise, it could cause delays in the progress of the clinical trial, of course. As I mentioned, there are phase-dependent requirements for quality documentation. And for clinical trials, the focus is on risk aspects. It's about the nature of the product, state of the development, or the clinical phase. It's about the patient population, the nature and severity of the illness, and the type and duration of the clinical trial. As Keith showed you in the last presentation, you have the CTD, which organizes the documentation, and it's exactly the same type of documentation that is requested for clinical trial, but much less detailed. In an MA application, the product will be very widely used in patients, and in a clinical trial, there's, of course, much fewer patients. And for an MA application, it's expected to have state-of-the-art in the documentation. But this is different for a clinical trial. The focus is on risk aspects. I'll speak about the EU guideline on the quality requirements of investigative medicinal products and the related questions and answers. The European guideline is called Requirements to the Chemical and Pharmaceutical Quality Documentation concerning IMPs in clinical trials. And the link to it is given here. This guideline of course, there's a guideline in the Commission, which has a name, something about the detailed information requested, but it's not really detailed on quality. But this guideline from the IMA, this is gives the guidance on the requested detail, the required detail of information which is needed for a clinical trial. It has been developed, it's a CHMP guideline which was developed by the Quality Working Party, and it came into force in 2006. And a couple of years later, after some initiative from industry, some question and answers were developed. It was some selected topics that where they felt that some more guidance was needed. And for instance, how to set specifications for impurities. I will come back to this later. And both these documents are available from the IMA webpage. The European guideline presents requirements for the investigative medicinal product itself, and also for modified authorized comparator products, and for placebo. For the modified products, this is relevant in most cases where you use blinding. Then if you have a tablet, then you have to maybe encapsulate it. And that's maybe the most common example of a modified comparator product. And in such cases, it's very important to demonstrate that the bioavailability has not been changed, and it's done by comparable dissolution profile versus unmodified product. And for all these three kinds of products, there's a requirement for GMP manufacture. And GMP is the topic for the next presentation. The guideline is, we can say that it is of an illustrative nature. It's not exhaustive, and it does not give detailed requirements for all kinds of relevant products. It's more general and illustrative. With increasing complexity of a product, then increased level of detail in the documentation would be relevant. For instance, for innovative or complex technologies, there may be a need for more detailed data to be submitted. And for sterile products, sterilization procedures other than terminal sterilization, then sufficient information on the manufacturing process and its validation is needed to be provided. So sterility assurance is important also at this stage. In general, there's significantly less details requested for clinical trials than for a marketing authorization application. But still, it's important to keep a future MA application in mind. I will give some topics here, but I will come back to more detailed examples in the next slides. Regarding qualification of impurities, it's a trend that the level of impurities will decrease with the development of the synthesis. So when you go from non-clinical studies to phase 3, there will be or to the MA, you will have a decrease in the level of impurities. And it's also important to consider the degradants occurring after reconstitution of the product, if that's relevant. Because those impurities may be different from the ones you see, which is synthesis impurities, and maybe also at a different level, at a higher level. And also, if the route of synthesis is changed, then other impurities may occur. So maybe, the change of a route of synthesis may cause some problems in qualification of impurities. So it's good to keep that in mind. So also keep in mind that APIs, where the composition of the API itself or its properties may vary, then it's very important that the batches for commercial product must be similar to the batches used in the clinical trials. At the clinical trial stage, then specification limits may not have been set, but it's still important to monitor all relevant parameters. Maybe for some kinds of specifications, they are not important in the clinical trial itself, but it's still important to just to monitor them and keep them for the future MA application. At the MA stage, at that application, then it's very important in the documentation that the differences between clinical versus commercial product is compared. And it should be, of course, as similar as possible, or sufficiently similar. This regards the formulation and the composition, and also the manufacturing process. I will now come to the examples, and I will start with examples where the level of detail is different in different phases in the European guideline. Regarding specifications for impurities, for phase 1, it's only requested to have tentative upper limits. But for phase 2 and 3, the specifications should be adjusted to the state of development. And in general, the limits should be supported by impurity profiles of batches of API that have been used in non-clinical and clinical studies. You probably know about the very strict ICH requirements for levels of impurities, and this is not requested at this stage if this kind of justification, as I just mentioned, is presented. And regarding genotoxic impurities, this is a very good example where there's very different requirements between clinical trials and marketing authorization, and also very different requirements depending on the duration of the study. It's for me a little surprising that the safety working party, they are normally very strict, but for a very short clinical trial, they allow a very high level of genotoxic impurities. And that can be seen in a question and answer document, which is has been prepared by the safety working party, and which is available on the IMA webpage. When it comes to validation of analytical procedure, for phase 1, it's only required that it's given some confirmation of suitability of the method, that it can measure what it's intended to measure, and also a summary of the acceptance criteria and parameters to be validated. But for phase 2 and 3, it's requested to have validation results for all relevant parameters. And I would say that this is relatively similar to what is requested for an MA application, but of course a much less elaborated report. Maybe a summary, some kind of summary may be sufficient. But results should be presented for all relevant parameters. Regarding stability of product, for phase 1, the stability studies should be started, and it should be given a commitment to continue the study during the clinical trial, and it should be given a justification of shelf life. For phase 2 and 3, in addition, it should be given results for accelerated and long-term conditions. And I think that it's not very much stability data that are requested for also for phase 2 and 3 studies. It's not stated anything about the minimum duration of minimum amount of data in the guidelines. So it's just some data. And very often you have more data from the active substance than for the product, and this could be used in some way as a kind of supporting information. But of course, also a justification of shelf life should be given. I will give some examples of the need to keep a future application of marketing authorization in mind. And this is most important for the quality parameters with potential clinical relevance. For tablets, which are very often applied for clinical trials, including modified release formulations, there is no requirement to set a limit for the solution rate. However, this should of course be monitored with a relevant test. And a more special example, if a very complicated active substance, such as a polymer, is used as a drug substance, it's important to monitor all relevant characteristics, such as molecular weight distribution, in sufficient detail in clinical batches. It could really be a lot of testing that needs to be performed to give a relevant characterization of such a very complicated substance. This may also be important if such a polymer is an excipient that is important for release of an API. And also for starting materials for synthesis, some planning may be useful at the clinical trial stage. Of course, there's very different requirements for detail in a clinical trial versus MA application. For clinical trials, less detail is expected in the description of the synthesis than for an MAA. But for a clinical trial, where critical steps have been identified, a more detailed description should be given. So also at the clinical trial stage, you should consider which would be the relevant starting materials for an MA application, in order to avoid to have to change the route of synthesis at a later stage. And also consider how the relevant starting materials will be provided later. And I've also included some examples on what kind of information is important to include. So little trivial examples of what companies sometimes forget. So it's important for stability testing to include relevant parameters that may change during storage, also including appearance and degradation products individually. And to provide justification of specifications for impurities and degradation products. Not forget to give information and results from validation of the analytical methods, and sufficient information on description and validation for sterilization by filtration. And also, many forget to also give information in relation to any dilution of the product before use. It is very common for some injection products. And then I would like to thank you for your attention.